sharing this.
Cool. Okay. Yeah.
Perfect.
Thanks, Emily. So welcome everyone to the August Recon Community Meeting. Today we are joined by Tobias Wenzel, who is an assistant professor at the Institute of Biological and Medical Engineering at the Pontificia Universidad Católica de Chile, PUC. He obtained his master's and PhD both at the University of Cambridge, I believe, in the Department of Physics, and he continued with his postdoctoral research at the Department of Chemical Engineering and Biotechnology. I think we missed each other by a couple of years here, I think. And he then held a Marie Curie fellowship before joining IIBM late in 2020. So his group now is dedicated to advancing open source hardware and software for high throughput biological research, with a mission to develop research grade tools for applications in bioimaging and microfluidics that can be locally fabricated and adapted, so it can empower labs worldwide, particularly in regions that are fairly underserved. So today, Toby's talk will be on local fabrication, global impact, on Latin American open hardware for bioresearch. So I really should just pass it on to you, Toby, to take it away.
Thank you so much for the wonderful introduction. I'm very excited to be here. And as I already mentioned informally before, that my motivation to be here, well, I would like to be also more actively involved in the network, but I really want to bring together these different communities that we're part of. So we're really pushing the openness, open research, really open science, not in the sense of open access publications, which is also important, but of really open physical objects running a biology lab in presence with all the things that are there, instruments, reagents, and so on, as an open science platform to empower sort of local communities and for them to be more innovative and more unique and more appropriate, so to improve research quality also. So I think we all have that goal in common, and I'm happy to tell you a little bit about our initiatives, and in that process, hopefully link those different communities a little bit more. Of course, yeah, the focus today is not necessarily on reagents, but more on instrumentation. And I know that there is a big overlap, and there are many other people in the network who are also thinking a lot about instruments, even though this is not the core focus of this network. But I still think it's good to connect these different initiatives that are going on, because there are a lot of common goals, even if the challenges are not exactly the same. So with that, first of all, jump in with a bit of a motivation of why we really build hardware ourselves. So, and why to do it as open source hardware, which is still quite unusual, even though I'm really glad to see this movement grow really fast. And there are more and more examples out there, but it's still not the status quo, right? So often we have to justify what we're doing. And so why do we build our own hardware? And the most important answer is to do novel, high impact science. So, I mean, this is more, why do we build our own instrumentation? Maybe not necessarily why do we do it as open source hardware? So it's to answer questions that we can't answer yet in science, we often need new technologies. Or maybe we just need different kind of technologies adapted to our local circumstances. But in order to do better science, we need our own instrumentation. And then dissemination is really important for us. So for me, it makes little sense to build or to develop interesting methods that then almost nobody can apply afterwards. So if you're in the method development, for me, dissemination is absolutely key. Like, what's the point of doing something that nobody can replicate? So sharing these things is so much more impactful when you develop it especially as low-cost open source hardware. And it's super important for reproducibility. It still surprises me sometimes that it's still quite common that people publish interesting data, and in the method section, they just say, this was recorded with a custom instrument that was done in our workshop. And I mean, that's not science at all. Like, you must say how you did this. So it should be enforced, but many times it's not yet. But I think it's an important part of reproducibility. And then there's maintenance, which is a huge topic for places like Chile. So when you are not there where most of the equipment fabricators come from, and you don't have as much science going on around you because you're in another location in the world, it's not one of the main hubs, then it's really difficult to maintain instrumentation. So not only to get it, but also to keep them running and to fix them without, a whole semester of downtime, for example, where the entire research group cannot progress. So this happens quite frequently in Chile. And so if you build your own instruments, and especially when there's a low-cost open hardware with local fabrication, you can adapt and repair them yourself and quite fast. And of course, we really want to do, and I think this, I like to think that we're also in the same boat there with the Reclone community. We really want to create this virtuous cycle of local innovation. So if you have more capabilities of understanding and managing and adapting these things locally, you can get to good solutions and also companies and knowledge innovation and many kind of benefits for the region. Okay, so those are the main motivations. And I will jump into the definition again as well, because just to make sure we're all on the same page. So the open source hardware definition is already quite old by the Open Source Hardware Association, which is a quite North America focused but global organization. And so they define open source hardware. It's a bit longer, but the main part is this. Open source hardware is hardware whose design is made publicly available so that anyone can study, modify, distribute, make, and sell the design or hardware based on that design. And the sell part is quite important because it's different to open source software. So you can have open source software that is not free for commercial use, but there's not so with open source hardware. And then there's the open science hardware movement, which is specifically for using open source hardware to drive science. It's a fantastic community. I think it's also quite closely integrated with Regosh, but where it's not, it's definitely a good thing to check it out. And especially, I wanted to add that link. Sorry, I didn't have Wi-Fi on the plane. So there is a very good forum. So if you Google it, you can quickly find it. It's very active. So this is a fantastic resource. I think there are many forums out there, but when it's not actively used and really housing a community, that's not very useful. But in this case, it's really fantastic. So if anyone wants to get going with open hardware in a biological context also, there's a great community that will respond to messages and questions and so on, and to share news. So this is something important to sort of keep in mind as a background for this presentation. And then one of the important reasons also why I'm presenting today is really that I'm leading this project, the Latin American Hub for Bioimaging through Open Hardware. So this is a big project financed through the Sean Zuckerberg Initiative, and that we're housing here at the Institute for Biological and Medical Engineering in Chile, together with two of my colleagues, Pablo de Razaval and Vicente Parot, that have a little bit of a different focus. So Pablo's mainly working on open source magnetoresonance imaging, and Vicente mostly in computational microscopy of neurons. And yeah, I mostly drive this open hardware context, but we will also see some of the instrumentation that I'm developing in my group and the microfluidics that we do. So this is a really great opportunity because we want to really push this. So especially in Latin America, but globally everywhere, I think, but especially here, I see a unique opportunity for open hardware and also open reagents, because it is logistically and financially very difficult to get access to common research reagents and instruments in Chile, in Latin America, really. So even if you can find the finance, which is often a hurdle, it's much more expensive to bring it in, and there's maintenance and logistics and different operating conditions. But in contrast to Africa in particular, there is a much better base infrastructure. So there are more funds available. There is more know-how available in good universities. 3D printers are everywhere. So I just think there's a good baseline to build on. But at the same time, there's a lot of need. And so we really want to push that. But to do this next to starting to be a professor is really challenging. So this project really made it possible for us in 4 years. So we have just over 1 more year to go in this project, to really hire a fantastic team also and get a lot of help to really organize a ton of things. That I'll jump to what we do. So we run a lot of workshops, hands-on workshops, where people learn how to build instrumentation. And then we'll basically give the instrumentation sets away So the participants apply with the motivation of what they would like to do with that in their research. And then they come, we in the workshop, they just use the documentation in a way, but we're there to give them some context and help when it's needed. So they assemble the instrument so that they also know how it works and use it. And then they bring it to their research labs and start using it for their research. So they get the set from us, which means that we only assemble relatively cheap instruments, of course, because we can't give away really expensive ones from the funding, but still, I think it's really exciting. And we've already done a number of workshops, and there are more to come. So we already had 2 on a strobe illumination microscope that we designed and built here in Chile. Also a low-cost laser illumination for microscopy that was developed here. Of course, we always build on other open source projects. So there are always components in there or certain sub-solutions that maybe other people have already published online or even in a paper. But then we take it some more further. On the power bioimage analysis, so all this course information and code is also available online on GitHub. Then we have one for LED illumination for fluorescence microscopy coming up, and another one on open MRI, sorry. So these are hands-on workshops that we do, but they're of course also for smaller groups and of course only for Latin American participants, unfortunately. So sometimes we do get people that are interested from other regions, but unfortunately we can't use our funds in this context to bring them in. But of course we always develop our resources for the whole world. So I'm very happy to support people to rebuild things, even if we can't fly them in and give them something for free in these workshops. And we do a lot of seminars also, similar to Reclone. They're a little bit less regular, but overall maybe even more per year. And these are focused also on open hardware. So we kind of interview, or we have presentations, scientific presentations of very cool open hardware developments around the globe, mainly related to bioimaging. So yeah, maybe I should clarify this bioimaging context. So the CZNI fund comes from the bioimaging program within the John Zuckerberg Foundation. And so there was a focus on bioimaging, but since we also do microphysics and microbiology and molecular biology in my lab, a lot of the tools go beyond classical bioimaging, and also a lot of these things are needed also for imaging. So it's all of an ecosystem really, but there's a focus on that. Okay. And recently we started to also add training videos. So this YouTube channel was a little bit of a surprise to us. Actually, initially, we just put the seminars up there because we could, but actually it turns out it's very difficult sometimes for people to really make the time to be there and present. But this has generated a lot of interest. So some of our seminars have ranked up to, I don't know, 80,000 views or something like that. So really, really a lot of attention online. And so we started providing more this asynchronous material. So these training materials that you don't necessarily have to come to a workshop, but anyone in the world can really watch that at any point. And especially the training videos we do in Spanish to make this more accessible to the Hispano-American community, let's say. And this is a community that doesn't exist. So the question is also how to reach all these scientists that might be interested. It happens very often in Latin America that people collaborate a lot internationally, but they collaborate mainly with sort of Europe and the US, where there are maybe better known scientists with also more resources. So it maybe makes some sense. But there are also very interesting people to collaborate within the subcontinent. And most people don't even know each other, and very few networks really exist. So this is also something we want to address, and it's difficult to get to these people. So we think that social media might be actually a good way, because then sort of everybody uses YouTube, so they might naturally find this content. And so we're trying to build a community through this, and so far it's going quite well. And so yeah, we started these tutorial videos and also videos on how to use open hardware to really perform like a scientific experiment. Okay, so check that out. And we also, of course, share a lot of 3D models that we design. We have a fantastic technology fellow in our team who's designing a ton of 3D printed parts, and we share them on printables and also on Thingiverse. A lot of interesting sort of pipette holders and stuff, but also syringe pumps, our microscope designs. Of course, the real documentation is often not here, so we share the parts, but then we link to the design files and the detailed documentation where necessary. And that brings me to the CAD files. It's really, really important when you design something to really share the actual design file, not just, for example, a printable file, because these printable STL files, for example, they're very difficult to modify. And there's actually a fantastic paper on this, that it's important for reproducible research to share these CAD files, these 3D design files. This is not, we didn't participate in this paper, but it's worth reading. And so we share all our design files. Unfortunately, it's not easy on this platform. We use Onshape, which is really fantastic, but it's not so easy for you to search for all the designs that we made. So I just see this in my login, let's say. But all the designs individually, for example, are linked to the printables pages and so on. So there, if you're interested in a model, you can find a link to the design, even though you won't see all of them from us like in one page like I do here. And we use Onshape also because it's unfortunately not open source. I wish it was. I think CAD programs, 3D like engineering design programs, there are open source alternatives, but they still have significant drawbacks compared to commercial ones. I think they're coming along. They're getting better and better. We're not yet at the point where I say we can totally jump on that with my group. And importantly, Onshape is the only software that I know where you can really collaborate in real time and also make forks and versions and merges in 3D design files. So that's a really important function. So it works like, like Git, basically like GitHub or even like Google Docs or so. So it's really fantastic for collaboration. And it's just online and you don't need to install anything. And it's free to use as long as the designs are openly available for now, but it's not open source. So another really important part is that we prepare a lot of detailed documentations, and we use Git building at this stage. So, well, some of you might know that I think in the early days of the open source hardware, open science hardware movement, I should say, I was a little bit known as the documentation guy, maybe. I also built a documentation tool, DocuBricks myself, but we actually don't use that anymore now. But we use Git building, to which we also contributed with the code. Because that allows version control and can be hosted directly on GitHub. So I think it has some important advantages. And a lot of the modularity and layout is anyways inspired by our previous software. So that is available here too. So this is really a step-by-step guide how to put together these instruments. And it's important because for us, it's open hardware if you just upload the design files and an open license, but open hardware is not enough. So we want bio users to be able to understand the design and build it. And for that, you need very detailed documentation because a lot of them do not have a lot of technical training. And we want that to be accessible for this community. So another thing that I always recommend is that you actually use other open resources. Don't just start from scratch, because you can, and contribute to those, improve their documentations, point out issues, and provide training material. So in particular, something that we've engaged with is, well, the open flexure microscope, which is everywhere. Everybody knows it. It's really popular. So we also have an automated class set, for example, that we use in undergraduate teaching. And we also did a workshop around it for some scientists. And also we made the first Latin American image data science with Napari course, which was online, and all the materials are available online. So Napari is a Python-based image analysis software environment. So most people still use Fiji or ImageJ for image analysis, very popular. It's still, I mean, I don't have the right numbers at hand, but it has an insane amount of plugins and implementations already available. So it's really still the default of the community. But all the modern tools, it doesn't have the capabilities to really work well with large data sets, 3D images, and also all the new machine learning things that are coming up that are just gaining so much importance. They're all sort of developed for Python, and so they don't work with ImageJ. So it's a little bit of a still the status quo, but it's a dying out dinosaur. So the new way, the future is Napari, basically. And so, but many people haven't worked with it yet. So we made a lot of tutorials there, how to use that for image analysis. And then, of course, we can do that with microscopy data sets that come from the open flagship microscope. Okay, so yeah, we share all our resources on GitHub. So there is a GitHub from the LibreHub project, but a lot of the instruments that we're developing are also directly on my lab GitHub. So there you can find all of these different resources, so documentations, design files, and so on. Now, I want to talk a little bit more about the why and how and the region. So for me, it has been a really interesting experience to move to Latin America. So I'm originally German. I spent almost 7 years in Cambridge in the UK. And so I've lived in different countries, and then Germany again, and an international organization, and then coming to Chile. And so a lot of people engage in hardware sharing, or even if it's reagent production sharing, I guess, because of accessibility. But there are important differences in what is really accessible when you are in a lower resource and also a little bit off the beaten track sort of country. So somewhere where not the major trade routes go through of high-end equipment, or So there are a number of aspects to it. So I actually wrote everything down in an article that already has received a lot of attention. And so I will only touch on some things really briefly now. But if you're more interested, I just suggest that you actually read the original article. So I tried to find a couple of data points also, because there's little evidence still in this new field that this is really a good way to access technology. So it makes sense to us, but it's difficult to evidence that to, let's say, the funder community. And so I looked for some data, and I found something really interesting. So if you look at all the public, on the left, you have this diagram. So the box represents all the publications that are published in this 25-year time frame. So there are 58 million publications, citable documents, that were published across the world from the Web of Science. And this is the distribution of the countries, how many papers come from which country. So yeah, it's not a surprise that the United States makes up a huge chunk, for example. And then you also see that Africa, for example, has only a tiny contribution to the overall publications, but also Latin America is very small here in the corner. But if you look at publications that mention Raspberry Pi or Arduino in the methods section, so these are much fewer papers. They're just 13,000, but still, it's not so small either. Then you can see that a lot of the lower resource countries around the world actually improve their representation among published articles quite a lot. And this is great because these low-cost tools and digital fabrication and so on, this is not only interesting to low-resource countries, right? So this is interesting everywhere. So a lot of interesting publications still come from high-end institutions that generally publish the most. But now the question is it really a help to these other countries? And I think this preliminary data set sort of really shows that yes. It's difficult to quantify, because you could put something like open source hardware, 3D printing. I did that also in the search and tried to find, but it's tricky because you can't be sure that they really use it in their project. So there are a lot of articles that talk about 3D printing as a fabrication method, but they haven't used 3D printing to make something that they used for an experiment. So it's maybe not such a good keyword to represent this, but Raspberry Pi and Arduino, even though the Raspberry Pi itself is not open source, but if you use it to implement a scientific experiment, it shows you that these kind of accessible tools are being used in science and the literature. So I think that's very encouraging. Of course, we need much more success stories. And then there's another thing. So when I arrived here, I was like, this is unexpectedly hard to do high-end science here and to build setups. And I was used to, I don't know, every department having their own professional workshop, machine shop, an electronic technician and so on. And of course, I've been spoiled in some of these institutions. But here it's absolutely absent. I mean, it's incredible. Like there is basically none in the country, not even talking about one in each department. Well, there is one machine shop in Chile, but it's been a little bit difficult to actually get something done there. So So I've looked at different data. So here's a lot of data from the UN and from the World Bank and so on. And it's interesting if you plot the different countries. So it's not only about the money that you have to spend, but here I try to plot something else. And this is the researcher density on the y-axis. So how many scientists do you actually have per population in your country? You know, how densely spread are people with this kind of know-how that you're working on? And on the bottom, I try to find another interesting, so it's kind of how to access complex tools. And a good indicator that I could find is the economic complexity index. So that tells you basically how technologically complex and advanced are the products that are being made in the country. And you can basically see that there is different kind of quadrants that I've indicated here. So there are these countries that have a lot of scientists, like Australia, for example, has a lot of scientists, but doesn't make very advanced products. So they're basically also a basic mining society. I mean, I'm simplifying it a little bit now, yeah. But they have a lot of scientists. So you do have a very strong scientific community. It's easy to find good scientists with know-how, but not necessarily to find, high-end machine shops. Yeah. Then, of course, there are the countries, a lot of European countries and US and Japan, some ones in Africa. Not Africa, sorry, not at all. But in Asia, sorry, I wanted to say. They have a lot of scientists and they also develop a lot of high tech in the country. So these are, so there it's easy to do this kind of development. And then there are countries that produce high technology, but they don't have a lot of scientists, like China, for example. So in China, it's really easy to get, I mean, it depends, of course, a lot where you are, but in China, there's also a lot of diversity. But generally, there's a lot of access. It's very easy to get almost anything done kind of close by and very fast. But maybe if you're trying to use this in a scientific context, it might be more difficult to encounter the know-how, for example, to contribute to that, how to use it, in which way, what for, but not difficult to do it. But a lot of the countries that I'm focused on, Latin America and Africa, more low resource countries, they're all down here. So they neither have a lot of scientists in the population, and they also don't have a lot of high-tech fabrication methods. So this tells us that if you will try to make something locally, you have to work with simpler methods, simpler components, and so on to be able to get this done effectively. I'm actually not as slow as I imagined with this, but I love talking about this. Sorry, I have to hurry up a little bit. Okay, and according to this, you can realize that not all open source hardware is born equal. So I've created these 10 kind of stereotypes of different types of open hardware project. These are just examples that I'm highlighting here to emphasize the idea behind it. Because now that more and more open hardware is actually being produced and published, I realized that not all of it is that suitable for local access to technology that can be maintained locally and built in advance. And I think this is a contribution towards an international discussion. So I'm not saying this is the ground truth for always, but I find that perhaps these 5 different parameters are particularly important in the context of biosciences. Yeah. So of course, cost is always important. So if it's cheaper, that's easier. But local fabrication is also something super important that I think we share with ReaClone, but that generally is it's not a very important topic that people talk about when they talk about accessible designs. I think it's so important that as much as possible of the device can be really fabricated locally. And adaptability, that's super important. Of course, you want to use it for science, you want to use it for something else in science, you want to adjust it to your local circumstances. Then something that I find also quite important is how a biologist, how someone without a lot of technical know-how would be able to implement it, but also to contribute to its further development. So this is a big gap that often happens in innovation and in companies. So the people that design the hardware and they use it, they're not the same people. And if we can bring these closer together, and if users can become developers, we will see very sort of powerful designs to emerge that just work much better in real life. And also openness. Of course, it's not so easy to quantify, but here I mean it to say how much let's say, design files and material are shared that is really modifiable and open. So, for example, in microscopy, it's very popular to just publish open microscopes, as in it's basically an assembly list, a bill of materials, and a documentation how to put something together of, let's say, Thorlab's components. So these are all high-end scientific components that are fabricated somewhere in the US and Europe. I don't know, maybe they also fabricate some in Asia, but I think mainly there. And then so you have to buy them all. They're quite expensive for a context of Latin America. You know, you also always spend a lot more money, at least 50% more, because of all the shipping and import fees. And it takes quite long. But these systems are very adaptable because they're very modular, and a biological user can easily put it together, understand it, and contribute to the development. I mean, with easy, I don't mean in 5 minutes, yeah, but if you're really working on this as a PhD student, for example, you can make a contribution. But the number of design files you share is really low, so it's just the list of what to buy, yeah. But if you also have software and you have CAD models to share, like 3D files that you can modify and so on, then more and more material gets passed along that enables you to work with. So the open flexure microscope, for example, I put it into the sweet spot. So this is the kind of thing that's relatively low cost. You can make almost everything locally. It's very adaptable. It's user-friendly for biologists. And well, this is not about user-friendliness really, but it's about they can contribute to the development. It's easy to understand overall. And there's a ton of material of openness that's shared. So all the design files are open, the code, and of course the documentations, what to buy. But there are many others that I'm not going to go into detail now, including here's the open wetware, like open enzymes. I've included them here as a category. I would like this. So this is a bit of a time snap point. Also, I think at the moment it's still not so easy to make yourself. And the whole production pipeline, I think there's still gaps, like things that we need to develop and share to advance this field. So that's why I didn't get sort of perfect scores from me yet in this assessment. Yeah, but I think it's a super exciting, even more future-facing area that I hope in the future will become even more accessible. But I think it's already fantastic, and I definitely wanted to include it. So usually in open hardware, we include sort of all these things that are physical, including reagents. Okay. So yeah, I already talked about these 5 dimensions. So basically with this discussion, I want to lead up to this point. So doing something that's open hardware is not enough. How am I doing in time? Half an hour is almost over. Yeah, I have to hurry up a little bit.
No,
but I'm
worries.
also
Tobias, feel free to talk a bit longer. It's fine.
Okay, thanks. So so what I realized, and maybe this wasn't so clear to me when I moved to Chile, also because now I'm leading a team doing this, so that really helps to clarify things, but also to write about it and so on really helps to clarify thoughts. So I had a certain way of doing it before, and that made sense to me. But then you realize other people are doing it differently. Why is that? What are their assumptions? What really works here? And there's nothing like really immersing yourself in this region to understand the limitations and so on. So really, doing something open source hardware is not enough for accessibility. So what we need is appropriate technology. Yeah. So this, and here is a definition that I like. There's several, but they come down to a similar direction. I'll read it out. A technology is appropriate when it is compatible with local, cultural, and economic conditions and utilizes locally available materials and energy resources with tools and processes that can be maintained and operationally controlled by the local population. Of course, in this definition, it's sort of quite extreme and sort of purist, but I think we have to go in this direction. And this is what when we talk about local fabrication. So this is why in a lot of designs that are developed in Europe and the US, they are maybe appropriate for those places. But we are in a very different economic context. We're in a very different infrastructure context, and as I mentioned, with the industry also, if the complexity of product fabrication is missing here, then we can't use the same kind of precision components, that they won't be available locally. So all of this makes it more difficult, and so it's important to think about the development of technology not just as open source, but also as making it more appropriately. And I think this is not black and white. So especially working a lot with optics and microscopy, I mean, it's very difficult to make the lenses yourself or the optical filters. It's not impossible, but even if you could, it might not make sense. So you might always have to bring some things in, import something, but the less that is, and the more appropriate parts there are, the more accessible the overall thing becomes. Yeah, so I think it makes sense to go for that, even if it's a partial approach. Okay, so local fabrication, as I said, is important, and also availability of parts. So we normally try to design things from very standard components, like the open flexure microscope, like a Raspberry Pi, standard screws, and so on. So or some industrial components, some machine vision cameras that we use a lot. They're really fantastic. And they're much cheaper than sort of scientific equipment normally, and they have similar performance. And they're just much easier to buy around the world. And this local fabrication development, also being able to develop the hardware locally, makes it much more appropriate for the context around the globe. And so 3D printing is an important part as local fabrication, and it has this double advantage. It's not only local fabrication, it's also digital fabrication, which means that there are more designs that you can share and adapt. So it makes everything faster. And so we try to design our setups mostly with digital fabrication so that other people can make it in the world anywhere quite quickly and cheaply. And for that, we design the parts to work with digital fabrication. So I do not recommend that people just take the metal parts they're used to, for example, in their scientific setups, and then print them. You will almost always get an inferior result, so it won't be as good if you just print it, because it hasn't been designed for that process and for that material. That doesn't mean that you can't print something that's as good, but if you do, it probably looks different. So what I'm trying to say is that it's good to design things so that they work well with printed objects and not just copy. And so here, for example, I developed this optical slit from 3D printed slides that you put together, and so that's an alternative. For example, this high-end slit. Of course, it has different pros and cons. And here, for example, we also just use a standard screw, whereas this uses a micro screw. So you can also buy these micro screws individually. And I haven't calculated this through, but I don't think it makes that much sense, because these screws you probably also don't really get locally. So if you just buy the micro screws and then sort of incorporate into this, then you might as well just buy the whole thing, probably at a slightly higher cost, but then everything is well tested. So in this case, the performance is lower because of the sort of standard component of the screw, let's say. So you could make it better, but that might not be justified. So you have to see what trade-offs you need. But the good thing is also that in a place like Chile, we can just easily make this in like an hour or so. So we say, oh, we need a slit, and then we have it. This is just a razor blade that's in there. And so this is really fast and easy. And time is also an important component, because it takes several hours of administrative work to order something internationally, and it might take a month or so for it to get here. And then, a slit like this will end up costing over $200 here. And so for this, we do it in an hour. It's basically for free. And that can make a substantial difference if you need more and more of these components in your setup. And for many applications, it's just good enough. But you can also have things that are better. But in this example, I'm just discussing it. Okay, so we develop a lot of high-throughput bioanalysis hardware as open hardware, and not only instrumentation, but methods. So we do these two things in my research group, and we want to share them. But again, for the methods, it's very important to us that other scientists around the globe can also use these methods in their research. And for that, often instrumentation is an important part. And so we're working on this sort of 3D printed open source hardware light sheet microscope that's in collaboration with the UC2 team in Germany also, but we have sort of our own version here, and a microfluidic droplet sorter. And we use a lot of 3D printing when possible to make these setups. And we're also working on some even more advanced setups that include sort of open source femtosecond lasers and so on. And they, at some point, that can also become quite expensive. But we're always trying to lower the cost significantly compared to most commercial items while at least maintaining the performance, sometimes exceeding it. Or just enabling it for other applications that don't need that much precision, for example, and therefore can be much cheaper. Like this light sheet, a lot of light sheets cost at least $500,000, for example, and this one costs just $2,000. Okay, so I would love to talk about all these in detail also. Feel free to talk to me about this or write to me. But this is just a brief introduction of what we kind of do. And I want to go into one example in a little bit more detail. So this is our open source strobe illumination microscopy stage, which we have particularly developed for droplet microfluidics. So you can do other things with it. It's a microscope. It's actually more of a macroscope. So in this case, you actually want to have quite a large field of view. The image quality is not so important. But since we modified a mechanism from the open flexure microscope, it's compatible with all their optical developments. So it's very easy to just put in better optics, for example, depending on your needs, and also other positioning systems. But in this case, you just want to have a pretty compact and low-cost setup that is really robust. So you can put a big microfluidic chip on top and move it around, and you can do all the sort of liquid connections. And when it spills a little bit, it's easy to clean. So that's really practical. And when you generate these microfluidic droplets, they move very fast. So for example, we generate 8,000 droplets per second. And so with a normal camera, you just see a line, like a blur, because they move so fast while you take one image. So in this stage, we synchronize a bright LED pulsing with the camera. And through that, we get like beautifully still images of these droplets. And this is equivalent to cameras that cost, $10,000 to $30,000. So normally all microfluidic labs need one of those. But we don't have one, for example, even though we do microfluidics all the time, because we don't need it. I mean, this works beautifully. And it's just a Raspberry Pi camera. And then also we have these 3D printed pumps that we advanced a little bit. And so altogether, it really fits into this framework. And yeah, of course, a lot of electronics. And altogether, it really fits into this framework of appropriate technology that I was telling you about. It's also quite modular. So there is this strobe module that we always use, but we also already have developed several temperature controllers and pressure and flow controllers, all part of this like Raspberry Pi web server, Python-based kind of ecosystem. But yeah, the strobe one, we already have organized several workshops, and so now already the strobe stage is available in several countries, including 2 in Argentina, 1 in Mexico, and 4 in different groups in Chile that are starting to use this in their research to do new advanced workflows. And because it's open source, it's really easy to adapt. So you can just easily integrate some tube holders, or if you don't have access to a laser cutter, you can just 3D print everything, or you can integrate other components like a better objective and XY positioning stage and so on. So that's all thanks to sort of the open hardware philosophy. And so with that, I want to pretty much finish up with the motivation of why microfluidics, because I think especially for the Reclone community, it's really interesting. Microfluidics is an ever-growing, it's not new, it's kind of the field is 40 years old, but it's been really complex to use for advanced biological assays. So it really took a long, long time for the field to develop and mature and have the different offerings and materials well tested to be able to do these. And it requires normally a ton of very expensive instrumentation, also know-how, which is why these high-end design system designs are usually not used in this open source community, but they're very powerful. And so I just want to explain that a little bit here, because also thanks to the different instruments and methods that we develop, I hope that more people in the Reclone community can use that to advance, optimize enzymes, test activity levels of their cell-free reactions or whatever it may be. So what you can do with droplets in particular is, so you miniaturize experiments. You also need less reagents, even though when you can make them yourself, you might not be so limited with reagents. But you also get less contamination. So if you do very precise assays like single molecule or single cell assays, contamination is always a big issue, but not so much in microfluidics. And you can do single cell experiments. And so in general, you work with these microfluidic droplets. So basically we have a chip where a cell suspension is encapsulated with a flow with oil. So oil and water don't mix. So you generate these droplets. They're stabilized with some surfactant. And this is with one of these high-speed cameras. So you really generate 8,000 droplets per second here. So it's extremely fast. And so in an hour or so, you can make millions of these little compartments, and they're one experiment. So it's like a well in a well plate, let's say, one droplet. So you do these high throughput experiments, for example, on directed evolution, and these are usually quite small. So this is like a human hair in comparison. And they're particularly great for interactions. So that's why we focus on biological applications of this in my lab, where we use interactions of cells, because you trap also the metabolites in there. So these are things you cannot do with a flow cytometer, and you can really see how cells interact. Yeah, should wrap up. Yes. But this could be great also for optimizing the activity of certain enzymes. You can have producing cells with cells where you want to see the effect on, or you can use biosensors and so on. You can use all of that in the same assay to select like single genes that express certain enzymes, for example. So here are just some tips, but I'm not going to go over it now. How you can implement this philosophy in your workflow of design and sharing so that they are more appropriate. And I also wanted to tell you that the Journal of Open Hardware that already has the community-run journal, and we just migrated to a new platform so it becomes Diamond Open Access. So now you can publish your designs there, including when it's about reagents, free of charge. So there's no fee to publish, but it's still open access. So with that, I thank you again. Sorry for running over quite a lot. So if you want to come to Chile and be part of the team, there are definitely opportunities for that. And thanks for listening.
Great. Thank you so much, Tobias. There's so much information that you've condensed into your talk. So I just want to open the floor to folks if they have any questions, and we can stay on for a little bit longer if people are available, but recognize that if you do need to leave, feel free to do so as well. So any questions for Tobias? While waiting, I want to thank Scott. You've been super helpful in posting some of Tobias's lab's links into chat. Sibeli and I have been in the background also curating a lot, so I've just posted a big message out there in chat. Feel free to save it and look at those links yourselves later. To me, this has been super fascinating to see how you've kind of blended the open hardware and the open software and into open biology stuff also into here. I would love to pick your brains more about your paper and how you measured it, essentially how you scored each of the different software. But perhaps that's a question for another chat for later, I guess.
Scored software? I'm not sure if I fully understand the.
Sorry. So when you're showing your diagram of the types of open hardware and how
right.
use it there, but I could also read your paper and ask you more on that.
Yeah, it's a start of a conversation. So I think it's just useful to know that I think I would have probably just said that a few years ago. Would have said, any open hardware is good. And I think it's still true. Like even the ones that now I think are not the most useful, it's so much better that people start sharing whatever information, even though it's not like all the way what I would like. But it's also important to recognize that I think people do this, they share this, and then they're not aware that is not going all the way. So maybe they think now everybody can do it. And then they might be surprised why nobody in Latin America and Africa replicates it. And so I want to start that conversation. But yeah, they're sort of different. Because also people think, I don't know, open hardware works badly and is cheap, for example. That's also another stereotype that people might have, right? But it's not at all true. I mean, there's like super expensive high-end material, including, all the electronics from the particle accelerator of CERN is kind of open source, and it's not cheap or low end at all. And in fact, it's not necessarily something that a biologist can easily implement either. So there's a whole spectrum. We have seminars of building your own electron microscope with open source and so on. So there's some quite high-end instruments. It's a really active community around magnetoresonant imaging and so on. These are very, very high-end instruments. In open microscopy, there are many instruments that cost several $100,000 just in components to build and so on. So there's the whole spectrum from not good enough for research, just education, to really high-end materials. Thanks for posting those links. That's really helpful also to the GOSH forum.
Yeah, no, I think Scott's really nice. We've been trying to curate that as you talk. I know Joseph had just said that he's got some questions he wants to talk to you about, but he needs to rush off, so he'll definitely get in contact with you about that more.
Okay, well, that's because I overran.
No,
Sorry about
it's
that,
fine.
but he can just get in touch and we can talk separately.
Definitely. Yeah, and it was interesting to know, how it's true. We have a fair few of the open DNA collections that are available, but we do need to do a bit more research into, how many people have actually, because one of the things we talk about is that we've shared our collections with over 500 researchers in 50 countries around the world, but whether people have actually made use of it, how they've made use of it, these case studies, these are things that we want to actually just get the numbers on to show that it's
I think it would be
accessible.
so valuable. I mean, this is also something we try to do with the open hardware, and many times it's really challenging to get this information. But this is really gold, I think, because it's still open. It's still early times. There are more examples, but there's very little proof that this really helps research. So even if it's still individual stories, but if you have several of them, and then there's still more hard evidence than what's there now. And I think this is really something that funders need, because they say, even if they think, oh, it's a pretty approach, but like invest seriously into sort of proven approaches, and so we don't know that, like, prove to us that this works, and so we need to bring that proof, and then we'll also receive more funding.
Yeah, that's always the aim. We always need a bit more support to try and keep these initiatives going.
Another thing maybe that I would like to mention that I didn't include it in the presentation, but I did post it already on the Reclone forum somewhere, that what I think about reagents that I need in the lab is not just enzymes. And so in fact, especially for bioimaging, you need a lot of different dyes to stain biological cells. And there are some really great chemists at Genelia in the US who develop stains, and they're very good at that. And they said that they would be very happy to host people from different, especially sort of underserved communities around the world, chemists that would be willing to synthesize these in different countries, so that we can do like local production of stains. I know this is a little bit different from the Recon focus, but I think it fits quite nicely into this ecosystem. So if you know anyone, it would be good to start building a community of chemists who can support the biology community in that way.
No, definitely. Feel free to post more about the forum and then also share it, because me personally, I don't know as many chemists, but I'm sure we can start coalescing all these different open communities together. Yeah, and also kind of a side note on that, there was a open source hardware meetup of different communities just a couple of days ago. But if you're interested in finding out more about that, Tobias, I can also connect you with the folks who organized that. It was actually organized by GOSH. Yeah, it's also always nice to see in these communities. And yeah, stains are huge. I don't know as much. Jose, would you like to interject anything more on that comment?
Well, I think they're just very similar to, yeah, maybe, I mean, if Jose can unmute, it will be even better. But just want to say that they can be as expensive as enzymes, and also don't have such a long shelf life and so on.
Interesting. Okay. Yeah, Jose probably not, I can't unmute, I can see, but he's posted that they have a project in microscopy, and it is a big limiting factor here. And yes, a supply chains issue, as we all know as well.
And similar to Reclone with like the off-license enzymes, I think similar things are true with stains. So stains are also very expensive. So I have no idea how to synthesize this. I mean, I haven't. Apparently, Jenny has made with a group member. They have made some evergreen, was it? So apparently that they made a huge batch, so that was great. But what the chemists tell me that it's really like not that difficult to synthesize, and so one of the reasons, or not that expensive also, but one of the reasons they're so expensive is, I guess, intellectual property. So so that's why the prices are so high. But the good thing is that, for example, in Latin America and Africa, I'm pretty sure that a lot of these things have not been like patented. So this means that even the proprietary stains, we could synthesize them ourselves.
Guess it'll be good to
And I think
how
also
this is
some
done.
off-patent ones.
Yeah,
You
definitely
have a question,
more
Jose?
opportunities.
Scott
Yes,
has to
open.
rush. Oh, thank you so much for the encouraging feedback. I just try to share a little bit across the community so that we connect better. Because I think that we're all emerging communities with maybe not exactly the same needs, but we definitely think we need to be connected and drive in the right direction. And Jose says, can you comment on how you make your case in Chile when the metrics are often about patents at the universities here? So I'm fortunate that, well, you probably all know Fernand Ferrici, who has the office next to me, is in the same institute. In fact, I knew about this institute from him before I even started looking into where to maybe go in Chile and apply here. So we're almost alone, but not completely alone in that effort to make this case. So the Católica, where we are, it's a private university. I mean, it's sort of semi-private. So in Chile, it's kind of, if you talk about private universities, you usually talk about other ones. So I think they talk about this university more as a traditional university, one of the first ones and the big ones that is very sort of culturally linked in, but it's still private. In the end of the day, I still want to make money. And this patent topic has become more and more important, and they're quite traditional and conservative in many ways. And we're not, let's say. So we always arguing with them about this, but I think that also the people in the technology transfer office, I mean, they do listen to us, and then they say that it makes sense to them too. I mean, of course, part of their job is facilitating all this patenting, and it is sometimes a metric that counts. And more for grants, you're also being asked how many patents do you have, and not how many open projects have you published. I mean, that doesn't show up. However, if you do publish them in a scientific journal, then that still counts, right? But I think also about impact. I mean, the universities also in the country, they are thinking also about impact metrics, revising them a little bit. They do like societal impact too, which is generally difficult to measure. So I think even though they advocate for it, I think they don't have a good way yet of rewarding it or including it in sort of, let's say, personal promotions or something. But you can always make that case in the text, and so it depends a bit on the academic units. So I think we always say, like, wait, there's more than patent, and they know about it. So that's what I would say. But maybe on a country level, there's so much more work to do, and we always could discuss this also in the microscopy community. It's just is that nobody can handle this alone, but we all need to lobby a lot more, sort of, to the governments and then to the general public, in fact, that science matters and it needs to be taken care of. Because in Chile, we are in this sort of weird position that it's not a poor country anymore. So we're sort of off most of the global subsidies that exist, or there are a lot of programs like Wellcome Trust and some other funders, the Gates Foundation, that only invest into really low-income countries. And so we're not that. But the government doesn't invest anything in science and innovation. I mean, not anything, but they're very, very, very low. So in terms of Chile, where they invest less than half of their GDP as in percentage compared to the Latin American and Caribbean average. So not comparing to Europe, but it's saying, of the money that they have, they put nothing into science. And so we're very poor in the science here. Not as poor as my dear colleagues in Argentina, who are really struggling at the moment. But we just have to make that case, and not just from one community, but all the scientists together, that it matters. and how to get there. And I think it's not just about patents, of course.
Great, glad that you could answer Jose's question there. Are there any more questions, or shall we thank Tobias for such a fascinating talk and let you guys get back on with your day? I'll leave it a beat. Okay, I can't see any more voices or things in chat. So thank you so much again, Tobias, for the great talk you've given us today. And we'll see
Thank you so much
guys
for
all
having
next
us.
time. Yeah, thank you. I'll see you next time, next month. Take care now, guys.
See you,
Bye.
thanks.